Hello, hello. Good afternoon, everyone. Let's get started. Today, we will be doing lecture 5, which is on metabolism. Specifically, we will be talking about cellular respiration. And if we have time at the end, we will take up the lab as well. Okay, and I'll also tell you about the test and then like a review for the test as well towards the end. Okay, so here we go. Write the word equation and balance chemical equation for cellular respiration. Indicate which reactant is oxidized or reduced. Okay, so the word equation goes like this: glucose, which comes from the food that we eat and is going to react with the oxygen that we breathe in. And after a bunch of reactions, many mini steps, we are going to end up producing carbon dioxide. And water. Of course, we will get some energy in the form of ATP as well. So the chemical equation goes like this. Glucose is C6H12O6. And we will be reacting with 6 oxygen molecules. That will release 6 carbon dioxide molecules plus 6 water. And under ideal conditions, we will produce up to 36 ATP. Okay. Now, this is a redox reaction. Redox reaction. Can someone tell me what does it mean by a redox reaction? Anyone want to tell me what redox reaction means?
It's oxidized plus
yeah, oxidation and reduction, right? Yeah, that's what this stands for. Yeah, but more importantly, a redox reaction is a reaction that involves the transfer of electron. Transfer of electron. Okay, so if something lose electrons, so loss of electron, okay, we call that oxidation. And a gain of electron, we call that reduction. Okay, so you can remember like Leo, loss of electron is oxidation, and Ger, gain of electron is reduction. Okay, so in our equation here, we will have electrons being taken away from glucose, and ultimately those electrons will end up on the oxygen. Okay, so glucose is going to be oxidized into carbon dioxide. Okay, oxidized into carbon dioxide. So this is oxidation. Okay, so we'll be removing electrons from it. The electrons that we're removing, they are high energy electrons. They have a lot of energy in them. Okay, and the oxygen is going to pick up those electrons ultimately. And in doing so, it is going to undergo reduction reaction. Okay, it's going to be reduced into H2O. Okay, so there we go. Any questions so far? All right, so this is a very complicated concept. It's very abstract for a lot of people, especially if you're learning this for the first time. So I will do a summary for you. Okay, but again, the full thing, it's probably better if you look at the recordings. But here I'm going to give you like an overview. Okay, just to recap some of the stuff. So this is cellular respiration. Okay, So cellular respiration has 4 stages, right? Can you guys type it out for me? 4 stages in the chat box. What are the 4 stages of cellular respiration? Yeah. We have the glycolysis. The next one is the pyruvate oxidation, followed by Krebs cycle. And finally, we have the electron transport chain, or the ETC. ETC. All right, so that overall equation that we saw, the glucose plus oxygen becoming carbon dioxide and water, that doesn't happen in a single reaction. It's like many, many different reactions. So in glycolysis alone, there are like 10 reactions, okay? And then like 3 reactions here, and then like 8 reactions here. So it's a bunch of mini steps that ultimately, if you cancel all the stuff that goes in and all the stuff that comes out, you end up having glucose and oxygen on one side, and then water and carbon dioxide and ATP on the other side, okay? So here is the premise, okay? We're doing an oxidation reaction. That means we're going to be ripping high energy electron away from the glucose. And that happens in the first 3 stages, right? So in these 3 stages, high energy electrons are released, okay, as glucose is being oxidized, okay? So we don't want to waste these high energy electrons. We don't want them to just like, flow away and, goes to who knows where. We want to capture them. We want to grab onto these high energy electrons, and we want to use the energy to make ATP. Okay, so in order for us to grab onto these high energy electrons, we are going to need to have some electrons carrier. Okay, so help me out, guys. What are some examples of energy carrier that we talked about in class? Okay, Moria said NAD, that's correct. NAD plus, that's what it's called. Not so much ATP. ATP is the end result, or we're gonna make ATP. What's another energy carrier? Yes, FAD. All right, so these are energy carrier. And what will happen is, if you add electrons to them, and again, this is an oversimplification, all right? The actual reaction is has more stuff in it, but we don't care. Okay? The NAD plus will pick up these high energy electrons, and it will get converted into NADH. Similarly, the FAD, after picking up the electrons, will become FADH2. Okay, so the NAD plus will be reduced into NADH. Okay, it's reduction because it's picking up electrons, it's gaining electron. Gain of electron is reduction, GER, right? Okay. So once they are reduced, these energy carriers will transport the electrons to the electron transport chain. Okay, they will deposit these high energy electrons at the ETC, and then the energy will be used to make ATP. All right, so that is the situation. Okay, so the energy carrier acts as like a taxi, right? And this is an unoccupied taxi, empty taxi. The passenger is like the electrons, so they pick up the electrons, they drive them to the ETC, drop them off, they will go back, pick up more electrons, and just keeps on going back. Right? It's like airport shuttle or whatever. Any questions so far about what's going on? Okay. All right then. So let's think about where these things happen, right? Okay, where do they occur? Let's draw our lovely cell here. I'm gonna put mitochondria. I hope you're drawing this along with me, right? Don't just sit there and watch and eat chips or something. Okay? Try to be more engaging. So there we go. There is my mitochondria. And the first step, the glycolysis, number one, takes place in the cytoplasm. Okay? That's where you take the glucose and then you will split it into two pyruvate. Okay. And then these pyruvate will go into the matrix, where step number 2 and number 3 will take place. Okay, 2 is pyruvate oxidation. Right? You're going to strip more electrons from it. And number 3 is Krebs cycle. Both 2 and 3 happens in the matrix. And number 4, the ETC, is going to happen in the cristae. And the cristae is the foldings, right, of the inner mitochondrial membrane. Okay. So let's take a look at what happens in the glycolysis. So glyco is sugar, lysis is breaking. Okay, we are trying to break the sugar. We're trying to split the glucose. So in glycolysis, we have two phases. Okay, we have the energy investing phase. Okay, you got to put in some energy first before you can harness it. And then number 2, we have the energy yielding phase. All right. So it's like investment. Right? You have to invest some money. You have to spend money to make money, right? People say, right? And that's similar here, right? Like you invest $10,000 into some stock, and then five years later, you make $2,000 from it. Okay? So you must first pay some money in order to gain some money. Of course, you can lose as well in the real world, but in the case of glycolysis, you would invest some ATP. Okay, so how many ATPs are we investing here? Anybody remember? Yes, thank you. 2 ATP. You must pay upfront $2. Two ATPs in terms of energy investment. So 2 ATP is used to, the easiest way to think about this is to energize glucose. Okay, make it a little bit excited, makes it a little bit unstable so that it's ready to react. Okay, so we'll do that. And then in the energy yielding phase, in the last 5 steps or so, you will be able to produce 4 ATPs. Okay, so in like glycolysis, you invested 2, but you produce 4. So overall, you're going to be able to get 2 ATPs. Okay, from this ordeal. And what also happens in the energy yielding phase is you will also produce 2 NADH. So high energy electrons will be released, like I have been saying. Some NAD plus going to come along, it's going to pick them up, and then we generate these NADH. Okay. Now I'm going to put a little star here. ATP, I never know if I should add an S or not, but whatever. ATPs produced in glycolysis. Uses what we call substrate level phosphorylation. Okay. So that's the mechanism. That's the method where ATP is produced in the glycolysis. Can someone tell me what does it mean to phosphorylate something? What does it mean to phosphorylate something? To phosphorylate something means what? Anyone?
To add a phosphate group.
That's right. That is exactly it. To phosphorylate something means to add a phosphate. Okay, so for example, ADP plus P becomes ATP. In this example, we are phosphorylating ADP to make ATP. Okay, so that's generally how you make ATP, right? You phosphorylate ADP, you add a P to ADP. The question is, how are you phosphorylating it? There are two types of phosphorylation in the context of what we're talking about. It could be a substrate level phosphorylation, or the other type would be oxidative phosphorylation. So I'm telling you, in glycolysis, the ATP are produced through substrate level phosphorylation. What does that even mean, right? Okay, so it means you are going to have, you know what, I'm just going to grab the picture from the lecture notes, because if I draw it, it's going to look too ugly. There we go. Okay, so substrate level phosphorylation involves, there's going to be an enzyme that facilitates the catalyzer reaction. There is going to be this here. This is a donor molecule. Okay, a donor, or like a fuel molecule. Okay, this is the one where we will be grabbing the phosphate from. Okay, we're going to be removing the phosphate from it. And of course, we need to have an ADP. So in the substrate level phosphorylation, you will take the P from the donor molecule, put it onto P, and as a result, we end up producing ATP. And of course, our donor molecule would have one less phosphate afterwards. Okay, so substrate level phosphorylation only accounts for about 11% of the 36 ATP produced. Okay. Any question? Now is the time to ask. All right. So overall, right, glycolysis, as I mentioned, essentially we will take glucose, and you will split it into two pyruvate. Okay, so sometimes you see people write pyruvic acid instead of pyruvate. It doesn't matter. For us, we're not going to make a distinction, even though they are a little bit different. Don't worry about it. Glucose. Oh my goodness. Glucose has 6 carbon. So if you split it into 2 pyruvate, how many carbons does each pyruvate have? Yeah, 3 is correct. 3 carbons. 3 carbons. Okay. There we go. So at this point, much of the energy is still locked in the pyruvate. You can continue to metabolize pyruvate, and then you get so much more ATPs and energy carriers from it. All right. So let's go back to our table here. Let's fill in the first row, stages in cellular respiration. The first step is glycolysis. Okay. Where does it take place? Well, I told you, right? It's in the cytoplasm. And the key products, we are going to have 2 net ATP. We're going to get some energy carriers, NADH. And we will have 2 pyruvate. All right. That is the first step. Hey, what's going to happen to our pyruvate now? Okay, so the pyruvate is going to enter the matrix. Okay, so again, we're going to have our mitochondria here. The inner membrane folds up into the cristae. Okay, so the glucose becomes the 2-pyruvate. Okay, and now these pyruvate will enter the matrix. Okay, and step number 2, pyruvate oxidation will take place. Okay. So we have our pyruvate. And 3 things going to happen to it. Okay. We are going to remove a carbon dioxide from it. Okay, so pyruvate has 3 carbons. And if you take away a carbon dioxide, the end product, which is called acetyl-CoA, will have 2 carbons, right? So only 2 carbons from the original glucose will be remaining. We will be releasing high energy electron from the pyruvate. So an NAD plus will come along and it will grab onto the electron and it will become NADH. Okay. And finally, since we have been getting so much energy from all these molecules, maybe it's losing a little bit of juice. So we want to energize it a little bit. We want to charge it up a little bit. Okay. So we're going to add something called a coenzyme. Coenzyme A. Okay. It creates a special sulfur bond that energizes the molecule so it is ready to react again. Okay. So 3 things happens and we end up generating acetyl CoA. All right. So how many times does this reaction take place for every glucose that we metabolize? 2 times is correct. Remember, each glucose produces 2 pyruvate. So each pyruvate will undergo this 1 time. Okay. So step number 2. Pyruvate oxidation. This takes place in the matrix. And we will get 2 NADH, 2 carbon dioxide, and 2 acetyl CoA. All right. Are we together? Any question at all? How is carbon dioxide removed from the pyruvate? Oh, well, it's a little bit complicated. We don't really have to know it, but on the pyruvate, like basically pyruvate, like there is a C double bond O group like that, okay, and essentially an enzyme will clip that off, and then it will become carbon dioxide. Yeah, it's probably a not a very good explanation, but that's the simplest sense without going into too much of the chemistry. Okay. All right, that is number 2. Okay. Do I have a picture of pyruvate in the lecture note? Let me see. I don't think I did. No, yeah, it's okay. You don't really have to know the structure. Okay, great. Number 3, step number 3. What's going on in step number 3? Step number 3 is the Krebs cycle. Krebs cycle, also known as the citric acid cycle, because the first product you get is citric acid. Okay. So what's going on? Well, this acetyl-CoA that we got from the previous step, it is going to, well, it's in the matrix, okay? So, and in the matrix, it is going to combine with another molecule. So that other molecule is called oxaloacetate, which has 4 carbons in it. Okay, so 2 carbons plus 4 carbon, that will give you 6 carbon. And the first product that you will get in the Krebs cycle is citrate. Again, citric acid, citrate is similar. Okay, we don't really have to know the difference. For those of you who want to know the difference, if you remove a hydrogen from citric acid, it becomes citrate. It's okay. Both is the same for us. So long story short, if you want a full movie, okay, watch the recordings. This is like one of those YouTube channels where they show you the whole movie in 10 minutes, right? I don't know if you guys ever see that. I hate it. Whenever I have a new movie I want to watch with my wife, and she's like, "Oh, I already watched the 10-minute version. I know the ending," and then she would spoil it for me. Anyway, this is like that, okay? It's the condensed version. So the citrate, it's going to undergo a series of reactions that will eventually regenerate the oxaloacetate. And in doing so, it's going to pump out a bunch of electrons. Okay, so we want to capture them, right? So we will capture them, and we will produce 3 NADH. Okay. We will also produce 1 FADH2. Remember, these are both of these are energy carrier. It's just that NADH carries more energy than FADH2. More on that later. And you will be producing 4 carbon dioxide. Oh no, sorry, my bad. 2 carbon dioxide rather, and then you will also be able to produce 1 ATP. Okay, and just like before, put a little star here, right? This ATP is produced by substrate level phosphorylation. Okay, you're gonna need to have a donor molecule, and you're gonna be ripping off the phosphate from the donor molecule and transferring it onto ATP. Okay, so that's 1 cycle of the Krebs cycle, 1 spin. Okay, 1 spin. Okay, so Adrian has, how does it produce 3 NADH? Okay, again, we are not concerned about the actual details. It's sufficient to know that as we break down the citrate, okay, along the way we will be oxidizing it, so we'll be removing electrons from it, right? And it will release sufficient electrons that you need to have 3 NAD plus to pick them up. Okay, so that's what it means. Okay, so like 1 NAD plus is not enough. There are so much electrons that you need 3 of them, and then you will need 1 from here as well. The last stage has 2 NADH. That's why. I'm not sure if I understand what that means. Like last stage here. Yeah, I mean, they're not really related, right? Like this is like, don't try to understand why there is 3 here, why there is like 2 here, and so on and so forth. Like they are kind of just the way they are, right? People discovered it, right? So that's kind of like what it is. Just kind of have to take it for granted, for our purpose, anyways. Okay, so 1 spin is gonna produce these much, right? So I want you to copy this down if you haven't already, because we're gonna do some questions on that later on. Okay, and in the PowerPoint, right? I show you something like this. This is if you Google Krebs cycle online, that's what it looks like, right? So it tells you exactly which step will the NADH be produced, right? But we just summarize it. We just say, okay, like 1, 2, and 3 in total. Okay? I remember like in university, we had to, the professor made us memorize the whole thing, like how to draw it and stuff like that. It's complete waste of my my memory. I remember nothing from that class. But anyways, all you have to do is remember 3 NADH, 1 FADH2, and 2 carbon dioxide per spin. Okay, per spin. So now go back to this very important chart. The third step is the Krebs cycle, yeah? Krebs cycle. And that happens in the matrix. Okay, so remember, one glucose split into two pyruvate. Each pyruvate will become an acetyl-CoA, and then each one of them will go through a spin, right? So essentially, one spin, if you roll it down, is 3 NADH. So that means per glucose, we will have 6 NADH. Okay. 2 FADH2, 4 carbon dioxide, and 2 ATP. Okay. And just to make sure that you remember, this is per glucose. All right. Does anyone have any question up to this point before we go into any more details? Are you sure? All right. So let's take a look at what happens at the electron transport chain. And let's write it out first. So in the electron transport chain, which happens in the cristae, right? The inner mitochondrial membrane folds into the cristae, right? And we're going to produce a lot of ATP. Okay, we'll say lots of ATP. Probably should say how many. So there's 36 in total. I already produced 2 here and 2 here, so that would be like 32 ATP. 32 ATPs. Okay. You will regenerate the NAD plus and the FAD. Okay, so after depositing the electrons in the ETC, they will go back to the first 3 steps and bring more electrons. And finally, we will generate the 6 water molecule. Okay. So we can see we have 2 carbon dioxides plus the 4 carbon dioxide. That accounts for the 6 here in total. And then we got the water. We have the 6 water, which is produced in the last step. Okay. There's just a few more footnotes here and there. These ATP, right? Produced via substrate level phosphorylation. These ATP, those are produced via oxidative phosphorylation. Okay. All right, let's do some polls. That is not the polls. There we go. Go ahead. All right, just 9 people answered. I prefer more people to pick something. Okay, so if you have been paying attention, I said most of the energy is locked in the pyruvate, right? You can look at this chart and you will see. Right? At the end of glycolysis, you have ATP, you have NADH, and you have pyruvate. The ATP is like the final steps. You got 2 ATPs, and then you're just going to be using them. The NADH you can use to make more ATPs, but the pyruvate, right? Look at the pyruvate and all these steps. You're going to be able to generate a lot more energy carrier and a lot more ATPs, right? So most of the energy will be still locked up at the pyruvate at the end of glycolysis, okay? Let's try the next one. Go ahead. All you have to do is look at the chart we did. Right? Look, ATP is produced here and here, and these are through substrate level phosphorylation. No ATP is produced in the pyruvate oxidation, and everything produced in the ETC is through oxidative phosphorylation. So the correct answers are glycolysis and Krebs cycle. Okay. Next one. Right. Anyone else? Okay. So NAD plus has more chemical energy than NADH. That is not true. After you pick up the high energy electron, it becomes NADH, right? So this one should be the one that has more energy. NADH has more chemical energy than FADH2. Now, I haven't mentioned this explicitly yet today, but it was in the notes. So each NADH has enough energy to produce up to 3 ATP, whereas each FADH2 can produce up to 2 ATP. Okay, so in that regard, this is correct. Jory shared a really good video that goes over all this stuff. Excellent. Okay, so thank you for sharing. It's in the chat box. You guys can take a look at that. Okay, part C, NAD plus can be oxidized into NADH. At first glance, this might seem correct, right? NAD plus becoming NADH, but you have to think about how it becomes NADH, right? In order to become NADH, it needs to pick up electron, right? And by picking up electron, it is going to be reduced and not oxidized. That's why this is not the answer. And finally, glycolysis can take place without NAD+. You need the NAD+ there to capture the electrons. If nothing is capturing the electrons, the reaction will not go. Therefore, this is not the correct answer either. Okay, so B is the right choice. Okay, let's go back to here. Let's take a look at what happens to all these energy carriers. Okay, we have been producing them along the way. What are we going to do with them, right? Okay, so this is going to be the step number 4, the electron transport chain. So this is going to be the outer membrane of the mitochondria. And then we're going to have the inner membrane that's folded up into the cristae. All right. Now, maybe a couple more things. This is the cytoplasm. This here would be the intermembrane space. The space between the two membranes, right? So intermembrane space. And over here, this would be the matrix. Okay? So like if we take a look at the mitochondria, we are basically zooming in on that section. Is it okay? Do people understand what I'm drawing and where we are at right now? If you have a question, please voice out. All right. Now, embedded within the cristae, we have hundreds and hundreds, thousands probably, of these membrane proteins. Okay, and then they form what's called the electron transport chain. Right? So basically, the electron transport chain, as the name suggests, will be responsible for transporting electrons. All right. So from step 2 and step 3, we created a bunch of NADH and FADH2, right? So the NADH will come to the first protein in the electron transport chain, and it will deposit the electron. Okay, remember the electron is like the passenger, right? Drop off the passenger, and now it will become NAD plus again. Okay, so NADH is oxidized back into NAD plus. So what's going to happen with this electron? The electron will be passed down the ETC. It's like playing a game of hot potato. This gets passed down to the next one and to the next one. As the electron is being passed down the ETC, what's going to happen is hydrogen will be pumped into the intermembrane space. Okay, from the matrix into the intermembrane space. Right? So if we keep on doing this, eventually we will have a lot of H plus in the intermembrane space. Yeah. Does that make sense? Yeah, someone had a matrix joke in the chat. I was waiting for that. It always happens every semester. So we have all these H plus here, right? Creating a high H plus concentration and then low H plus here, right? So the hydrogen really wants to come back in, right? Because it's want to go down its concentration gradient, but it cannot because the cell membrane, as you know, will not allow charged particles to go through. So there is a special doorknob looking thing. It actually does look like that, apparently. This is called the ATPase, okay? Or sometimes it's called ATP synthase, depending on where you read this from. and it provides like a channel for these H plus to go through. Okay, so as the H plus goes through the ATPase from high concentration to low concentration, it's like water going through a turbine. It turns it, and that provides the energy necessary to produce ATP, right? Okay, so we're gonna finally be able to take the ADP, we're gonna add a phosphate to it, right? Through this turning mechanism, we're gonna produce ATP. Okay, so ATP produced this way is called oxidative phosphorylation because you are phosphorylating the ADP through oxidation reaction, right? Oxidation reaction means you're losing electrons, right? You're transferring electrons. So that's basically what it means. Okay. Now, how many ATPs you can produce? Well, like I said, the NADH will supply enough energy to pump enough H plus to produce up to 3 ATP. Okay. If it's a FADH2, the FADH2 having less energy will deposit its electron lower in the ETC. Okay, so rather than at the very top, it's going to do it at the second protein, and as such, there will be less H plus being pumped into the intermembrane space. So with less H plus, you will be able to generate only 2 ATP. Okay, so the key point here is NADH can generate 3, FADH can generate 2 ATP. Seriously, nobody have any question after all that? Okay, couple more things. Now this ETC is like, it's like slide. Okay, it's like a slide at the park. All right. If you ever take your kids to the park, you will know there's always going to be like some kid who lingers at the bottom. Okay, and then if this is a good kid, they would not go when there is another person at the bottom. Of course, sometimes they don't care. They just go and ram their feet into them. But normally speaking, the next kid cannot go unless someone remove that kid from the bottom, right? Okay. So in a similar sense, once the electrons get to the last part of the ETC, something has to take the electrons away. Okay. It cannot stay there. If it's stuck there, then no more electrons can go. And the bigger problem is if electrons is not being passed down the ETC, we cannot harness the energy. We cannot pump the H plus. If we cannot pump the H plus, we cannot generate ATP, and that's a big problem, right? Okay, so like a parent come along and then take the kid away, right? So in this case, right, the thing that acts as the parent would be, anybody want to tell me what I'm about to say? What molecules needs to come to take the electron away? Anybody? You guys still with me? Yes, the oxygen must come. So the oxygen is the final electron acceptor. The O2 will come, it will pick up the electron, and it will get converted into water. So that's the final electron acceptor. Okay. Now, the electron gets passed on to the next protein and the next, and then eventually to the electron because of something called electronegativity. Okay, my other class told me you did not learn about this concept in chemistry. Is that correct? Maybe, maybe not. I don't know. The electronegativity means attraction for electrons. Okay, the higher the electronegativity means stronger attraction. Okay, so if you think about the ETC, if you think about the electron transport chain, okay, protein number 1, protein number 2, protein number 3, and then finally we have oxygen, right? As you go down the ETC, the next protein in the chain is always going to have a higher electronegativity. It must have a stronger attraction for electron. Right? So look, this one. If number 1 has the strongest attraction for electron, it will never let go. Okay, it's because number 2 is stronger that the electron will be transferred to number 2. And then number 3 is even stronger. So it's going to go over there. And then finally, oxygen is the one with the highest electronegativity. So electron will, be passed on to the oxygen, and then off we go. Okay, so understanding this is important. Okay, you must have something that is stronger in electronegativity that allows you to go on. Now, we'll talk about cyanide poisoning later. Cyanide, if you get cyanide, cyanide will get stuck to the ETC, and cyanide has even higher electronegativity than oxygen. So if you have cyanide, the electron will be stuck here, and even though you have oxygen, it doesn't matter. It will not get pulled away from the ETC. So in the presence of cyanide, the entire ETC is going to get clogged up, and we're not going to be able to make ATP. Okay, you will die essentially very quickly. So yeah. Okay. Any question? Okay. Let's go back to here. We'll skip this for now. We'll come back to it. I was just trying to save space because this chart does not fit on this page. So I just kind of swapped the order. So in this case, we're going to try to account for the 36 ATP that is being produced in the entire process. So let's start with glycolysis. Okay. Number of ATP through substrate level phosphorylation. How many do we produce through SLP in glycolysis? Anyone? Two net, that's correct. 2 ATP net. Okay, energy carrier produced. If you recall from the first page, we have 2 NADH. Okay. Now there's a little bit of a catch here with these 2 NADH. Okay, remember. Again, this is the mitochondria, and this is the cytoplasm, right? These NADH, these ones that are producing glycolysis, they are actually in they're actually in the cytoplasm, right? These NADH. Okay, and in order to get to the ETC, you have to enter the matrix. But NADH is too big; it's not going to be able to enter the matrix. So what happens is, there's going to be a FAD here, okay, and then the NADH will just kind of pass on the electron. Okay, so the electron will go to the FADH, the FAD, and then the FAD will become FADH2. Okay, so in the process, we lose some energy, right? Okay, no energy transfer, no reaction can occur without loss of energy. It's the second law of thermodynamics, right? So here, by doing this transaction, we cannot just pass the electron from an NADH to another NAD. Okay, it has to be something that carries less energy. And again, the reason we have to do this is the cristae is where the ETC happens, and the NADH is too big; it cannot go inside. All right. So what happens is the NADH, as I said, will basically pass the electron to the FADH2 in the matrix. Okay, and then these NADH2 will be in the cytoplasm, right? So ATP produced, right? So 2 FADH2 multiplied by 2, because each FADH2 can produce 2 ATP. So that will account for 4 ATPs in total. So from glycolysis alone, if you add up all the ATPs that we produce, that's going to be 6 ATP. Okay, I hope the numbers are making sense. What about pyruvate oxidation? How many ATPs are produced through substrate level phosphorylation? How many should I put here? That's right, none. There are none. Okay, no ATPs are produced this way. And energy carrier, we will have 2 NADH. Okay. And these two NADH will go to the ETC. We are already in the matrix, so we don't have to transfer them. Just multiply by 3. So that's 6 ATP. Right? So if you add it all up, we will have a total of 6 ATP. Okay, again, just to emphasize, ATP produced at the ETC is oxidative, right? Oxidative phosphorylation. Okay. All right, here comes the big one, the Krebs cycle. How many ATPs through substrate level phosphorylation? Yeah, per glucose, we have 2. 2 ATP. Energy carrier. Right? If you remember from the chart from the first page, 6 NADH, 2 FADH2, and that's it. Okay. So 6 NADH times 3, 2 FADH2 times 2. That gives you 18. And this gives you 4, right? Okay. So 18 plus 4 plus 2, that is going to be 24 in total. And if you add 6 and 6 and 24, you get your 36 ATP. Okay. If you're wondering, why am I learning this? I just want to, go into practical nursing. What does this have to do with anything? It's standardized curriculum for all the pre-health programs. So even if you go to a different college, you will still be learning the same thing. I know that's not really a comforting answer, but it is what it is. So there, you have your 36 ATP. So once again, I ask, anybody have any questions? Or we are crystal clear on this stuff? All right. Here's a chance for you to show me you actually understand the poll question. Go ahead. Anyone else? Only 8 people answered. We have 22 students here. Only 8 answered. What about the rest of you? Where are you? All right. Looks like that's as much as I can get out of you guys. There's the result. Let's see how to do this. It's very simple. 4 NADH. So each NADH can produce up to 3, right? So we would times 3. 4 times 3 is 12. And FADH2 times 2. 3 times 2 is 6. 6 plus 12 is 18. So the answer is 18. All right. Some of you chose 7. Actually, some of you choose like some of the other stuff. Are you okay now? Do you understand why it's 18 and not 17, 7, or 21? Okay, great. Here's another question. Right. Please try that out. And if you have an answer, you can type it into the box. Okay. You have to account for all the NADH and FADH2 as well. Okay. So do that and let me know. I'm waiting for some answers. Okay. We have people saying 30. We have people saying 60. Anyone else? You know what? Why don't we just go 90, 120, and then not sure, I guess. Okay. Let's change this into a poll. What if you get this question on the test? What are you gonna choose? Okay. The popular choice seems to be 60. Is that the right one? Let's find out. Okay, so what we need to do for something like this is we have to figure out one term. Okay, so one term, we have 3 NADH, right? 2 FADH2, sorry, 1 FADH2, and 1 ATP, right? This is from one term. You're like, how am I supposed to know that? Well, we did it, right? Over here, right? 3, 1, 1. So then, what happens is you will multiply this by 3, multiply this by 2. So that gives me 9, 2, and 1. So if you add up all these numbers, 9 plus 2 plus 1, that's going to give you 12 ATPs per turn of the Krebs cycle, right? So we're going to turn it 5 times. 12 times 5 is 60. That's the correct answer. You will definitely see something like this on the test, so make sure you understand it. Okay, there's a long comment. Hold on, let me see. The hydrogen that's being pumped from matrix into the intermembrane space, that would be active transport, correct? Yes, it's active transport, and the energy that is supplying this active transport is from the high energy electron, right? Okay, let's take a break first, and then we will we'll come back and continue. And in the meantime, I'm gonna launch a poll for this challenge question, and maybe you can give that a go. All right. See you in about 5 minutes. Okay, we'll resume at 1:40. Hello, it's me again. let's keep going. All right, so we have 6 people who've answered the challenge question. Why don't we take it up? Let me read it to you first in case you haven't. Hydrogen cyanide, HCN, is poisonous. It binds to one of the membrane proteins in the ETC and blocks the movement of electrons. If cyanide poisoning occurs, what would happen? Sorry, what would happen? What would happen to the pH of the intermembrane space? Right. So let's think about our picture here, the one that we did. Okay. So the ETC allows us to pump H plus from matrix into the intermembrane space, right? The energy is, of course, supplied by the high energy electron. So if you have cyanide stuck here, okay, and it's not allowing the H plus to go through, what happens is you will have less H plus in the intermembrane space because you're blocking this pump, right? Okay, by clogging up the ETC. So by having less H plus in the ETC, less H plus, that means it is going to be higher pH. Okay, so the correct answer is A. It's going to be more basic, okay, as opposed to more acidic. Does that make sense? I certainly hope it does. Okay, let's go back to our fill in the blanks. This is the matching the terms. I will just post up the answer. You can check later. We probably don't have time to fill that in. Let's go ahead and fill this. Let's fill this in. Okay. So we have 2 types of fermentation here: alcohol fermentation and lactic acid fermentation. So glycolysis, we have glucose, and then we get the pyruvate. Right? So pyruvate has 2 fates. Right? In the human body, it has 2 fates. It either goes to step 2, 3, 4 that we talk about, right? The pyruvate oxidation, Krebs cycle, and etc. Right? And that happens if we have enough oxygen. Right? But if we have low oxygen or not enough oxygen, then it's going to go an alternate pathway. It's going to go to fermentation. Okay. So glycolysis plus all these things, right? We call aerobic respiration because it can only occur with oxygen. Whereas on the other hand, the fermentation that happens when there is not enough oxygen, so that is anaerobic respiration. All right. So let's take a look at lactic acid fermentation, which is the kind that we do. Okay, when you're running at full speed, as fast as you can, top speed, you cannot maintain that forever. After maybe like 2 minutes, 3 minutes, then your muscles start to hurt, right? That's a signal that lactic acid is building up, and your body is not producing enough ATP to sustain that kind of movement because you don't have enough oxygen, right? So you will eventually have to stop. Right? So starting with glucose, we're gonna do glycolysis. Okay, and as you know by now, glycolysis will be releasing electrons. So we will have 2 NAD plus coming in, picking up those electrons, and it will become 2 NADH. Okay, never mind this 2H plus. We did not talk about it, so just cross that out. And of course, we will have 2 net ATP as well. So this thing that's being generated, that's pyruvate. Okay, and someone was asking earlier, right? Like in pyruvate oxidation, how do you produce the carbon dioxide? So you see, there's the C double bond OO, right? So something's going to take that, and that's going to pop out and become carbon dioxide, right? But that's not what's going to happen here because we do not have enough oxygen. So the pyruvate, instead of going to 2, 3, 4 steps, it will go and become lactate. Okay, you see it's missing an H here. That's why it's lactate and not lactic acid. Okay, so pyruvate is going to be reduced into lactate because it will pick up the pick up the electron from the NADH. And in doing so, it regenerates NAD+ so that glycolysis can keep on going. Okay, so the purpose of fermentation is to break down pyruvate and regenerate NAD+ such that glycolysis can continue. Okay. I mean, it's not fantastic because glycolysis can only produce 2 ATP, but it's better than nothing. Okay, it will keep you going for a couple more seconds and then you will have to stop. All right, and all that lactic acid that you generate, it burns, right? Like if you haven't exercised for a long time and then you started exercising, strenuously, the muscle will burn, will ache for the next couple of days, right? That's the lactic acid. All right, so that is lactic acid fermentation. Now, other organisms, they also do fermentation, but they do a different kind, alcoholic fermentation or just alcohol fermentation. Okay, we don't do alcohol fermentation because alcohol is like poisonous to our cells, right? We don't want to generate that. In bacteria, in yeast, they do alcohol fermentation, not because they don't have enough oxygen, but because they don't have the ETC and all the complicated enzymes needed to do the Krebs cycle and stuff like that. They don't have it, okay? And they're single cellular organisms anyways, so it's okay to rely on just fermentation. So just like us, they will have glucose and you will have glycolysis. That will produce 2 ATP. We have our pyruvate here. The NAD+ will pick up the electrons and it will become 2 NADH. And never mind that. Instead of taking pyruvate and becoming lactate in one step, they need to do 2 steps. So first, they will remove a carbon dioxide, right? Kind of like pyruvate oxidation, remove the carbon dioxide. And after you remove the carbon dioxide, it becomes something called acid aldehyde. Acid aldehyde. Okay. And aldehyde is something that has a C double bond O and an H on one side. You will learn that in organic chemistry next semester. And the acid aldehyde will be reduced into ethanol. Okay, so if you use yeast to bake, the carbon dioxide release from this process will help your dough to rise, right? And bacteria can be used to make drinking alcohol as well, right? Ethanol is the drinking alcohol. Not every alcohol can be consumed, right? The alcohol that you spray on surfaces to clean. Anybody know what that alcohol is? Right? Drinking alcohol is ethanol. What's the one that you spray with? What do we call that? Anybody? You must know, right? Everybody's obsessed with cleaning during the pandemic. What are those alcohol called? Really? Nobody knows? The stuff that you spray on surface and you clean, or you guys don't do that? People are spraying the grocery with it, right? No, those are propanol, right? Isopropanol, right? The ones that you buy from, so the rubbing alcohols, right? Those cannot be consumed. If you drink it, you will get poisoned and you will die. So ethanol is the drinkable kind. There's also methanol, right? The stuff that you put on to kind of soothe the pain of aching muscle. That also cannot be consumed. So there you have it, anaerobic and aerobic. Let's go up here. We still have one more chart to go. All right, this is just summarizing the information. So lactic acid fermentation examples, that is the muscle. Okay, muscle in human cells. When does it take place? When oxygen demand exceeds oxygen supply. Where in the cell does it take place? Cytoplasm. And we generate lactate. Okay. Alcoholic fermentation. That happens in yeast, bacteria, and it takes place like basically all the time. That's how they make energy in the cytoplasm. And they will create carbon dioxide and ethanol. Okay. Any question? Okay. Let's take a look at here. Any requests for the long answer questions here? I think most of these stuff I've talked about already. There shouldn't be any confusion, but just in case, anything? All right. Application question. The cyanide poisoning, we talked about this already, right? By blocking the ETC, the H plus cannot go to the intermembrane space. If we do not generate the concentration gradient, then you won't be able to produce ATP and then you die. Number two, speculate what would happen if toxin disrupts the integrity of the inner membrane, inner mitochondrial membrane. Okay, so similar concept here for number two, right? So you have a lot of H plus up here in the intermembrane space, and they will go through the ATPase to produce ATP. However, if you have a bunch of holes in your inner membrane, and all of a sudden the H plus will be able to leak back in. They don't have to go through the ATPase, right? So you are reducing the amount of H plus that's going through the turbine, that's going through the ATPase. That will reduce your ATP production. Okay, so depending on how severe it is, you know, it could be a pretty big problem. Okay, if a significant amount of H plus is leaked out instead of going through here, then you would have critically low ATP production, and that could not be good. Right? For example, if you don't have enough ATP production in the heart, then that could be lethal, right? So there you have it. I will post the answer for these fill in the blanks, of course, and you can check them yourself. But that is lecture 5, and this is going to be the last lecture of the unit test. Okay, so let me just pull up the calendar for your class so I can tell you what to expect. Where are we? Over here. Okay, so we are at week number 6 right now. And next week is engagement week, so there's no class. Right? You just take a week off. But make sure you take that opportunity to study a little bit as well, because when we come back, the test will be on the Thursday, and it will cover lecture 1 to 5, as well as your lab. And so to help you prepare for the test, our next tutorial will be on the Tuesday instead of the Thursday. Okay, so of course I will send out a reminder when we get closer to it. Okay, but it's going to be on Tuesday, 12:30 to well, 2 hours, right? Basically. Okay, but do study a little bit. I'll post a review sheet for you to do, and we'll be taking it up on the Tuesday, the week after next. Is that all right? Okay, so let's take a moment and talk about the Beetroot Lab. You probably have read my feedback already, but maybe it's a good idea for me to go through it with you. I mean, the lab was not done as well as I hoped it to be. Not sure what the problem was, but again, a lot of people seems to have not watched the video and they just search up answers online. It's very easy for me to see whether you are answering the question yourself or like whether you're copying it from another place, right? Number one, people copy stuff from online and then paste it here, and they don't even bother to change the fonts. Okay, so I see the fonts look very different. I know you didn't type it there, you copy it, right? So that's like a dead giveaway. And also the wordings are a little bit weird. And sometimes the answer that some of the students provide has nothing to do with what we are talking about in the lab. Okay, like we only change temperature, and then they start talking about using alcohol, changing solvent. Then I know it's not something that they come up with themselves, right? So that was a little bit disappointing. Someone asked, when will you post a review sheet? I'll post it like today, okay, after class, in the debrief. Jory said, to be fair, some of the questions you asked weren't in the video at all. Yeah, they are not, right? And not everything is stated explicitly in the video. Some questions do require you to think critically, for example, like number 4, right? And it's okay to do a little bit of research in the internet, right? I'm not saying don't do any research, but like you got to filter the information, right? Like you got to know what's applicable and what's not, and don't copy them word for word, right? In many cases, for people who did copy, I was able to find a website, right, that you got the information from, and I would have made a comment and told you not to do that. Could we get an example answer for the next lab? I feel like what do you mean like an example answer? Like for the example answer for the, I don't understand what do you mean, Sasha? Like answer some of the questions, to show you how to answer some of the questions? Is that what you mean? Like if you show how an answer would, well, yeah, I mean, I kind of did for this lab too, right? I explicitly type out the answer for the first question for you, right? And surprisingly, not many people use that. Okay, people still kind of make their own answers. All right. Anyways, let's take a look at this. Let's see. There's more comments. Some of the questions did require specific research. Like which one? Which one requires specific research? Jory, maybe you can let me know. So we know degree of critical thinking. Okay. Anyway, I'll take this up, and then maybe you'll know what I'm looking for. What is the purpose of this experiment? I told you already, right? It's to investigate the effects of temperature on membrane permeability. That's all. Okay. If you change the temperature, what's going to happen to the membrane permeability? Then you predict it in your hypothesis. Okay. So the hypothesis is increasing the temperature will cause the permeability to increase, okay, or decrease, or no change. You would choose one of those, and that would be your hypothesis. Okay. Where people lose mark is, if you say changing the temperature will affect the permeability. Okay, that is not specific, right? Like, how does it affect it? Does it increase it? Does it decrease it? What's the effect? You got to make that explicit prediction. Dependent variable is going to be the color pigment, the intensity of the color. Right? You can also say the percent absorbance, whereas the independent variable, that would be the temperature. Why is beetroot chosen for this experiment? Why not carrots? Why not tomatoes? Okay, so many of you said that the beetroot has the a pigment in it, right? Which is great. They do have the pigment, but you also have to consider, like, why not these things, right? They also have pigments. As you know, carrot is orange. It has beta carotene, and then tomato is red, right? It also has its own pigment. So why not? Well, to get full marks, you would have to tell me, number one, the color is very, very vibrant. So it's very easy to make a distinction. The carrot, yes, and the tomato, yes, they have their pigments, but it's not going to produce that rich color that is going to be detectable easily, right? Certainly not by eyes, right? And if you use a machine, maybe the difference is not it's going to be quite subtle, not going to be able to create a sharp difference. And number two, if you think about cutting the beetroots, right? It's going to be easier to cut the beetroot into uniform shape than you will be able to do it for carrot. Carrot is probably too hard to cut. And tomato, if you ever cut tomato, it's like very watery on the inside, right? So it's hard to get these uniform chunks. So those are some reasons why beetroot is more suitable than the carrots and the tomatoes. Most people got this one right. You will lose marks if you just list a bunch of stuff. So I have people who put down like, goggles, knives, gloves, aprons. What about them, right? Like you gotta explain a little bit. The test tube that is the control is the 20 degree. Remember, a control is something that you don't manipulate it. Okay, so 20 degrees close to room temperature in this case. That's the one that we will be using as a reference to compare all the other test tube to, right? That's the importance of having a control. It creates a baseline for all your other treated groups to be compared to. Okay, so many people miss this second part, and you will lose a mark for not doing that. What needs to be taken into consideration when cutting the beetroots? You have to make sure you say that they're uniform size, make sure they have same surface area, right? It should be taken from the same beet, right? All these are examples of standardized variable. That's the keyword I was looking for, right? You can change the temperature because that's the independent variable, but everything else should be identical, right? And to ensure everything else is standardized, you need to make sure that you're cutting them the correct way, the same size, everything else that I said. Next one. Why is it necessary to rinse the cut beetroots? Right. So you will have to remove the excess pigment that got leaked out from the damaged cells when you cut. And why is that important is because you again, it goes back to the idea of standardized variable, right? If you don't rinse it and you put it in, then the solution will begin as red already, right? Without any pigment leaking in, it would just turn red from all the juice that basically bleed out from the cutting, right? So we don't want that. Okay, we want all the water to be clear to begin with. That's why it's important to rinse them at the like after you cut them. This one, all right, to get 4 marks, you really need to tell me why not longer than 20, why not shorter than 20. If you do it for shorter, it's likely that you are not going to see a very big difference. If you do it for 10 minutes, for example, the color difference is not going to be there's not going to be as much contrast. It's going to be harder to tell the difference. They might all look very pale because the pigment doesn't have time to come out yet. Okay, and if you do it for longer, the opposite could happen. Right, they could all turn very, very dark, and also the temperature of the water will also start to drop, and you're no longer submerging it in 80 or 60 degrees. So for that reason, 20 degrees. The recommended time is suitable, not longer or shorter. Sasha asked this: did we find out the class average for the lab? I mean, I could find that for you, but a lot of people didn't hand in the lab. I must have like 7 or 8 people who didn't hand in their lab. So the average, even if I pull it, it's going to be artificially low because of all the zero, right? I'm not sure why people didn't hand it in. I don't know, maybe 10% was not worth it. Anyways, moving on. Describe the correlation you observe between temperature and color intensity. Very straightforward. You just have to tell me as temperature increases, the color intensity also increases. You don't have to explain or anything, right? This is just pure description. And in part B, the answer you give in the part A is an example of qualitative analysis. Explain what that means. Okay, so qualitative analysis means you are making observations with your senses, right? In this case, you are looking at the color intensity, so that's observation using sight. Okay, it's not using numbers. You're not measuring anything with any device. Can you give us the average without including those who didn't hand in? Yeah, sure. I'll do the calculation. Okay, I'll multiply by whatever and then divide it by whatever minus the 7, 8 people who didn't hand it in. I'll do that. Okay, remind me if I forget. In this experiment, how was the result measured quantitatively? Right, using a calorimeter, right? So many people put color emitter. Okay, it's not color emitter, it's calorimeter. Okay, I'm not sure why people put that. Maybe they heard it, or maybe was it auto generated from the caption? I don't know. Okay, but I also know you guys have your own WhatsApp group, right? You guys told me. So maybe one person said color emitter, and then everyone in the group just put color emitter, but it's calorimeter. Okay. Next one, describe the relationship between the absorbance measured by the calorimeter and the color intensity. The higher the color intensity, the higher the absorbance. Okay, so darker the color, higher the absorbance. The graph was generally done okay. You will lose mark if you didn't label the axes, for example. Oh yeah, I did not take off any marks if you put color emitter. Okay, like I just tell you what it was called, and then I still give you the mark. Okay, so like don't worry about it. Over here, you will lose mark for the graph if you didn't label your axes, or if you use the wrong type of graph. Okay, some people have like 2 lines instead of 1 line. You should have a single line where y-axis is absorbance and x-axis is the temperature. A couple more to go. At what temperature does the absorbance peak? I would accept anything between 60 to 80. Okay, you can see the graph kind of goes up and then it plateaus, right, after 60. So that's where it has peaked, right? It's not going up anymore. And this is probably going to be different if you are comparing it to another plant cell. Okay, a lot of people say since they have the same pigment, then the absorbance should peak at the same temperature. It's not about the pigment, right? It's about the membrane composition, right? We talk about this in class. If you have a membrane that has a lot of cholesterol, for example, then it's more fluid, right? So something like that is going to peak at a lower temperature, and then for some. Sorry, they had a snow day today, and I look outside. There's not a single snowflake. I don't know why they closed the school. That's why my kids are home. Anyways, so like it depends on the membrane composition, right? Maybe you have more saturated fat, and that will take a higher temperature to make it leaky, right? So depending on what it's made up of, the peak temperature is going to be different. 2 reasons why temperature affects membrane permeability. Most people got this. First one is if you increase the temperature, there's more kinetic energy, right? So then, you know, the phospholipids would start moving faster, and that creates more space between them, and things starts to leak out. And then if you really crank up the temperature, as we go up to like, 60 to 80, the protein starts to denature. And once they denature, right, the integral proteins, if you think about it, that's what holding the membrane together, right? So if those things starts to denature, then your whole membrane starts to fall apart, and that's when a lot of the pigments start to leak out. Conclusion, you will say increasing temperature will increase permeability, as evident by the increase in color intensity. Do you accept or reject? So you refer back to what you said at the hypothesis. You either agree it or not. Sasha said, yeah, it just said appropriate graph. Yeah, and the appropriate graph is the line graph, right? If you give me like a bar graph, I don't think I took off marks for bar graph. I would take off marks if you didn't label though. But some people have a graph that has like 2 lines, and that's not the appropriate graph, right? You wouldn't have 1 line for temperature or 1 line for absorbance. That's not how you represent the data, right? Okay, some people use pie graph. That's not an appropriate graph, right? So the most appropriate graph to represent the data would be the line graph, and that's what they did in the video, right? I believe. Number 4, the frozen beetroot. We talk about this in class, right? When you freeze it, right, ice crystal is going to puncture the membrane, right? The expansion of the water will rupture the cell membrane. So even when you bring it back to room temperature, the whole membrane is full of holes already. So everything would just leak out, regardless of the temperature. So even though we are doing it at room temperature, the absorbance is going to be very high. Okay. So I mean, if you have individual questions about your lab, okay, if you think one of the question is graded unfairly or whatever, it's okay. You can email me. You know, state your case, and then I will tell you what I think. You know, we can work something out. And a lot of people already email me. Right? They told me, you know, I think I should get more marks here and there, and then I explained to them why it was the case it is. So if you're concerned, right, please do send me email. But I hope you guys will do better next time. Don't get me wrong, like for the most part, people did a decent job. Okay, but you know, it's just overall is not as good as I hoped it to be. All right. So that's it. No class next week, and I will see you in 2 weeks. Enjoy your week off. And if you have any question while you're studying, please feel free to contact me. I don't have the average right now. I gotta find it and calculate it. I'll post it on the debrief. Okay, I promise. All right. See you guys later.